We'll go ahead and get started for today. We'll start by going over the homework. So is everyone able to see the homework? Okay, cool. Okay, so we'll start by going over the vocabulary words. So we're gonna start with helicase. Okay. So which of these definitions describes what helicase is? Right, it's an enzyme that opens up the DNA double helix. Right, so that's going to be involved with DNA replication. Okay. So during DNA replication, in order to start the whole process, what we need to do is we need an enzyme called DNA helicase, and what it'll do is it's going to basically unwind the DNA. Okay, unwind it, and in doing so, it's going to be breaking the hydrogen bonds in between the nucleotides on either strand of the DNA strand or of the DNA molecule. Cool. All right, next one. So we got DNA polymerase now. Okay. So how would we describe DNA polymerase? DNA polymerase. What do we think here? Right. So it's going to be an enzyme that that synthesizes DNA. Right. Which is sorry. So an enzyme that synthesizes a new strand of DNA that's going to be complementary to the previous one, or the parent strand. Perfect. Good. So DNA polymerase, when we're trying to replicate our DNA, we're going to need DNA polymerase to add the new nucleotides onto the new daughter strand of DNA. Okay, perfect. Next one is the leading strand. Okay. So which of these defines what the leading strand is? Right, the DNA strand that's synthesized continuously in the 5' to 3' direction. Right, so remember when we are looking at replicating our DNA, one of the strands is going to be replicated continuously, the other one is not. Okay. So the one that is replicated continuously in the 5' to 3' direction is the leading strand. Perfect. All right, good. Next one is the Okazaki fragments. So how would we describe what the Okazaki fragments are? So the Okazaki fragments are not a short strand of RNA. What would they be? Anyone have an idea? Yes, Kimberly's got this one right. So this is the DNA strand, okay, that gets synthesized in short segments, okay, or short DNA fragments. So remember, when we're looking at the leading strand, the opposite strand is going to be the lagging strand. Okay? But in order to produce the lagging strand, the DNA needs to be produced in short fragments. Okay? Because again, you're going to be going in the 5' to 3' direction. Okay? But as you open up the DNA on, as you continue to open up the DNA, you end up with new fragments getting formed or getting exposed. So you need to produce those new DNA fragments. Okay? And you have to keep adding fragments in one segment at a time. And so those would be the Okazaki fragments. Those are formed inside the lagging strand. Okay? So the leading strand is synthesized continuously. The opposite strand, the lagging strand, is going to be synthesized in DNA fragments. Okay? And so those are the Okazaki fragments. Okay. All right, next one is the primer. Okay. So what is a primer? What do we think here? Yes. So the primer is going to be a short segment of RNA, okay? And that is going to be found during DNA replication. Okay? So in order to start DNA replication, DNA polymerase needs to bind onto the primer. Okay? So it's going to be a short RNA sequence of nucleotides that's going to sit in order to help you begin initiating DNA replication. Right. Okay. Any questions on any of these first 5 definitions before we go forward here? Okay. All right. If not, go to the next one. So we got RNA polymerase. Okay. So what is RNA polymerase? Anyone have an idea? Right, Kimberly is good. So this is an enzyme that's going to help synthesize an RNA strand, okay, from a template DNA strand. Okay. So RNA polymerase is going to work very similar to DNA polymerase. The difference here is that RNA polymerase is going to be involved with producing RNA nucleotides to help you produce an mRNA strand, a messenger RNA strand. And so that is going to be used during transcription. So DNA polymerase gets used for DNA replication. RNA polymerase gets used for transcription, okay, to help you produce a new RNA strand. Good. Okay, what about a template strand? What is a template strand? Yes, so it's going to be the strand of DNA that's going to specify the complementary mRNA molecule, right? So when we're looking at a template strand, the template strand is always going to be used to help you produce an mRNA molecule in a complementary manner. So that is going to be used to help with transcription, and the template strand is also going to be used for DNA replication. Okay? So the template strand is going to be one single DNA strand that gets used to help produce either a new DNA strand during DNA replication or help you produce an mRNA during transcription. That's going to be the one that is used to help you form the opposite strand in a complementary manner. Okay, good. Next one is splicing. So what is the process of splicing? Yes, good job, Christopher. Right. So splicing is the process of removing introns and exons, or removing introns and then reconnecting your exons. Okay, and that's going to be found in your pre-mRNA. Okay, so remember when we're when we're looking at eukaryotic cells and they're undergoing transcription, there's going to be a bunch of sequences in there that are going to be useful, and there's going to be some that are not useful. Okay? So the ones that are not useful are what's called introns. Okay, those are the intervening sequences. And we do not want those, okay? So we want to try to pluck those out and make our mRNA as efficient as possible. The exons are the ones that are useful to help us produce our protein when we begin translation, okay? And so what we want to do is we want to remove the introns, okay? Get them out, remove them all together, and then just reconnect all of your exons together so you have a full mRNA, a full mature mRNA molecule, okay, that is going to be useful to help us produce various proteins. All right, good. So that is splicing. Okay, next one is tRNA. What is tRNA? Yes, so good job, Kimberly. So a tRNA molecule is going to contain a 3-nucleotide anticodon, okay? And so that anticodon on the tRNA is going to bind onto the mRNA in a complementary manner, okay? So for example, if you have an mRNA codon that says, ACG, okay, then the anticodon on the corresponding tRNA would be UGC. So it's going to have a complementary anticodon to the mRNA's codon. And so that tRNA molecule is going to bring a specific amino acid during translation to help you begin producing your mRNA molecule. Or sorry, your protein, your polypeptide or your protein. Okay. And the last one is the start codon. What is our start codon? Yes, good job, Yetzi. The start codon is the AUG sequence on an mRNA molecule. Okay. So the mRNA is always going to have a start codon, and that is going to signify the beginning of translation. Okay. And so it's always going to code for the amino acid methionine. Okay. So for example, the first amino acid in any protein is always going to be methionine, MET, okay, because AUG is always going to signify the start codon. That is going to be where you begin translation. Okay. Any questions on any of these definitions? I know there's a lot of definitions and a lot of kind of things happening here. Any questions on anything we've talked about so far? Okay. All right, so then we'll go ahead and move on to the next question. So question 2 says, which of the following is true about DNA polymerase? So we got A, DNA polymerase only works in the 5' to 3' direction. B, DNA polymerase does not require a primer to begin in the leading strand. C, DNA polymerase unwinds the DNA helix. Or D, DNA polymerase breaks down nucleotides to replicate DNA. Which of the following is true? Yes, A is correct. Yeah, so DNA polymerase will only work in the 5' to 3' direction. Okay? It cannot work in the 3' to 5' direction, which is why we end up with lagging strands and how we end up with the Okazaki fragments. Okay? It's only able to work in the 5' to 3' direction on the new DNA strand, okay? the daughter strand. Okay? B is wrong because, again, in order to begin DNA replication, you do need a primer in both strands. Okay? C is wrong because DNA helicase is going to be the enzyme that helps to break down the DNA helix or unwind the DNA helix, as we saw earlier. And then D is just wrong because DNA polymerase doesn't break down any nucleotides. Okay? So the only one that's true is that DNA polymerase only works in the 5' to 3' direction. Cool. All right, again. Next one. So question 3. So if a strand of DNA sequence reads 5' on the 5' end, we have A, T, A, G, C, G, C, G, G, T, A, T, A, or T, A, T, T, C. Okay. And then that's going to signify the 3' end. Okay. If this sequence is replicated, okay, which of the following accurately represents the newly synthesized complementary DNA strand? Okay. Which one do we think? All right, so I've got one person saying B and another person saying A. One of those is true. Okay. Anyone have an idea of which one? Yes, so the answer is A. Okay. So A is going to be true. So the DNA sequences are going to be the same here. However, what we have to also pay attention to is the 5' and the 3' kind of on our orientation here. So Jared got that exactly right, because of the 3' and the 5'. And so if our DNA sequence is going to read 5' on one end and 3' on the other, then the opposite strand has to begin with 3' on this side and the 5' on this side, okay? Because again, they're going to be anti-parallel. Okay? So if this is our 5' end, the opposite would be the 3' end on, oops, sorry. The opposite would be the 3' on the complementary strand, and then the 5' on the other side. Okay? And then you have your rest of your DNA sequence over here. Okay, that's how it works. Okay, so I'm glad this is all making sense to people. Perfect. Okay, any questions on this one? Okay. Again, you're just creating the complementary strand here. Okay, the only thing you have to also pay attention to is the direction of the complementary strand as well. Cool. Any questions on this one here? Yeah, so you just flip it essentially. So yeah, so Mario's question was, you just flip it? Yeah, you essentially just flip it. Okay, you make sure that you do create the complementary strands, and then you just flip the directions. Okay, 3' on one side and then 5' on the other. Cool. All right, good. Glad that one makes sense. Okay, number 4. So nucleotides are the building blocks of DNA. Which of the following is not a component of a nucleotide? Okay. So we have A, deoxyglucose, B, phosphate, C, deoxyribose, or D, a nitrogenous base. Okay. Which one is not a component of a nucleotide? Yeah, so Kimberly's got this one, deoxyglucose, right? Deoxyglucose is not really a thing, okay? So this is not going to be found in DNA. However, we do find deoxyribose, which is where DNA gets its name from. Okay? We have a phosphate, and then we also have a nitrogenous base. These 3 components will be used to help us create a nucleotide. Good. All right, next one, question 5. So which of the following is true about start and stop codons? Okay. So A, the stop codons UAA, UAG, and UGA end translation and do not code for any amino acids. B, the start codon codes for the amino acid phenylalanine. C, the start codon is GUA. Or D, different tRNAs carrying different amino acids can bind to the start codon. Yeah, so I'm seeing A, and that is absolutely correct. Right, so the stop codons are going to tell you when translation is going to end. Okay. The other thing that's going to be important about these stop codons is they do not code for any amino acids. Okay. So whatever the amino acid you produced before you reach the stop codon, that will be the amino acid that is the last one in your protein. Okay. B is incorrect because the start codon is always going to be AUG, and so the amino acid it will always be producing will be methionine or MET. Okay. Again, that kind of leads us into C. Okay, start codon is not GUA, it's AUG. And then the last one is also incorrect because you're not going to have different tRNAs binding to the start codon. You're only going to have a single amino acid carrying methionine to the start codon. Perfect. All right, question 6. So RNA polymerase reads the following DNA sequence on the template strand. And we have ATT, CGA, TAC, CGT. Okay. That's on the template strand. So what would the resulting mRNA sequence be? We'll take a look at those and let me know what you think. Right, what are we thinking? Right, we'd have the first one. Good job, Hasmi. So the first one would be correct. This would be our resulting mRNA sequence, okay? Assuming we're looking at the template strand. Okay, we'd have UAA, GCU, AUG, and then GCA. So you're just creating the complementary nucleotides to the DNA template strand. Okay. Why is C not correct? Okay, why is this one not correct? The one that has the T's in there. What is wrong with this one? Yes. So, Pasmina, yes, you kind of got this one right. So, in mRNA, okay, we do not have any U's. Okay. We don't have any T's. Okay, our T's get replaced by U's. Okay, and so our T's, our thymines, end up getting replaced by uracils inside the mRNA. Good job, everybody. That's the only reason. Okay, we don't have any T's in mRNA. We just have U's instead. All right, good. All right, cool. So question 7. So we have the following mRNA sequence: AUG, AAA, GCA, UUA, UGA, ACG. Okay, so after translation is completed, a polypeptide is created. So use the genetic code to help determine the amino acid sequence of this polypeptide. So I'm going to go ahead and just kind of zoom out here just so we can see all the options. Okay, so based on what we see here, and based on our amino acids, sorry, our mRNA sequence, what should be the amino acid sequence that we produce here? Right, so Jared has got A, and so did Hasmin, and that is perfect, right? So we start off with finding our first codon. So here we have AUG. So AUG is going to give us methionine, and that's going to be our first amino acid. Then we have AAA. Okay, so we have AAA over here. So AAA would code for lysine, or LYS. Perfect. Okay. Then the next one would be GCA. GCA would give us alanine, which we see over here. Perfect. Then we have UUA. And so UUA would give us the amino acid leucine, or LEU. Perfect. And then we have UGA. Okay, UGA codes for the stop codon. So we're not going to be producing any more amino acids. We're then done at that point. Okay. So once we hit the stop codon, we are finished. Leucine was our last amino acid, And so this would be our amino acid sequence. Cool. All right, good. Okay, that is perfect. So question number 8. So we have a tRNA molecule in the P site that releases the polypeptide chain it is holding. Okay, what will happen to this tRNA molecule? Okay, so we have something that's in the P site. What's going to happen next? So we have A, the tRNA will move into the E site and then be released from the ribosome. B, the tRNA will move into the A site and block incoming tRNAs. C, the tRNA will move into the E site and stay there for the remainder of translation. Or D, the tRNA will move into the A site and be released from the ribosome. What do we think here? Right, A is correct. So we're going to be having our tRNA molecule that's in the P site. It's going to move into the E site. Okay, and you kind of think of the E site as the exit site. That's going to be where it's going to exit the ribosome from. Perfect. Good. Okay, any questions on anything so far? Let's zoom back in here. Okay. So then we have question 9. So question 9 says, which of the following best describes the relationship between DNA, RNA, and proteins? So we got A, DNA is transcribed to make RNA, and then RNA is translated into proteins. B, DNA is transcribed to make proteins, and proteins are then translated into mRNA. C, RNA is transcribed to make DNA, and then DNA is translated into proteins. Or D, proteins are translated into RNA, and then RNA is transcribed into DNA. Which one is true? Yes, good job, Christopher. A is true. Right, so DNA is going to get transcribed into RNA, and that's going to be the transcription process. Once we form our RNA, the RNA gets translated into our proteins. Perfect. Good. All right, question 10. DNA and RNA have what in common? Okay, A, the number of phosphate groups attached to each nucleotide. B, the 4 nucleotides they contain. C, the type of ribose present. Or D, the number of strands, either double-stranded or single-stranded. What do they have in common? So I see one person said B. What else do we think here? Yes, so A is correct. The 4 nucleotides, sorry, the number of phosphate groups. Okay. So there's going to be 1 phosphate group attached to each nucleotide. Okay. Now, B is incorrect. The 4 nucleotides, okay, almost sounds like a band, like Kimberly said. The 4 nucleotides is not going to be correct. They're going to have different nucleotides. Okay. And so if we think about DNA, DNA's nucleotides are going to be A, T, C, and G. Okay. RNA, on the other hand, will have A, U, C, and G. Okay. So they differ in the nucleotides that they contain. Okay. So they're not going to share all 4 nucleotides. Okay. The second one is, or the third one is incorrect. Okay, the type of ribose is incorrect because DNA is going to be made from a deoxyribose, while RNA is going to be made from a regular ribose. Okay. So the ribose is going to be different here. Okay. And then the number of strands is also going to be different. And DNA is always going to have 2 strands. RNA will typically have just 1 strand of nucleotides. Okay. So the only thing that they have in common is the number of phosphate groups attached to each of the nucleotides. Okay. That is that. All right, and the last one, what type of bonding holds DNA nucleotides together? We have A, hydrogen bonding, B, covalent bonding, C, ionic bonding, or D, van der Waals forces. Okay. Which of those 4 holds the DNA nucleotides together? Yes, and absolutely correct. Everyone's got this one. This is hydrogen bonding. Perfect. Okay. Any questions on anything we've covered so far, or anything from chapter 9? I know there's a lot going on in chapter 9, so if there's any questions, feel free to let me know. Okay. All righty. If not, let me go ahead and clear this real quick. There's just a couple of announcements. So, first one is that exam 3 will be on Thursday. So exam 3 is going to be on Thursday. That'll be open at 8 o'clock in the morning, and then it'll close at 11:59 p.m. So again, you can take it anytime during that window, but it needs to be completed by 11:59. Okay. If it's not, it will be, you'll end up getting a 0 on the exam, and this is the last exam before the final. You don't want that to happen to you. Okay. So exam 3 will be this Thursday, same format, 40 questions. It's going to cover chapters 6 through 9. Okay. So chapter 6, again, covering mitosis and all the stages happening there. Chapter 7, covering meiosis and all the stages within that. And then what can happen if meiosis goes wrong. Chapter 8 was genetics. So there's going to be about 8 to 10 Punnett square questions. Okay. So make sure that you feel really good about the Punnett squares in terms of simple genetics, or sorry, simple dominance and simple recessiveness, as well as the non-Mendelian genetics. So codominance, sex-linked traits, incomplete dominance, and epistasis and things like that. And then chapter 9 going over DNA replication, the structure of DNA replication, transcription, and translation. And then the last thing we're going to be talking about today is how genes are regulated. Okay. So that's going to be on Thursday. So again, same format as always. Just be ready for that. Okay. Another quick announcement. So if you guys are in the Wednesday lab or the Thursday lab, okay, there was a glitch with the with last week's classwork. So classwork 421, which you see over here, and classwork 423, for some reason when people were submitting it, it wasn't submitting all the way, and I wasn't able to see it on my end. Okay. And so I kind of had to go around that. So I had to create a new classwork 421 and new classwork 423. If you have not already, complete these by the end of the week, ideally today. But if it takes till the end of the week, that's fine. These have the same question as the original ones. So if you want, you could even just copy and paste your answer if you feel confident in your answer, or you could just redo it. Tuesday lab, this was not a problem. So if you're in the Tuesday lab, disregard what I'm saying right now. But this is just for the people in the Wednesday lab and the Thursday lab. Okay. So if you're in the Wednesday lab and Thursday lab, please complete new classwork 421 and new classwork 423, even if you've already done classwork 421 and 423. These ones I'm going to end up having to delete, and so you'll not get any points for that anyway. Okay. So complete new classwork 421 and new classwork 423 if you're in the Wednesday lab or the Thursday lab. Okay. Tuesday lab, everything works fine, so disregard what I'm saying. Okay. But it's just for Wednesday and Thursday. Okay. Any questions on anything that's coming up? Okay. So if not, last thing here, I know this is kind of a ways away, but the final exam will be 3 weeks from today. So the finals are coming up. So that will be on Tuesday, May 19th. Okay. So the exam will be up on Canvas. I'll talk more about it after this next exam. I don't want you stressing about it now. But we're starting to kind of review all the material, starting to feel comfortable with the material that we've covered since the beginning of the semester. Okay. Okay. Any questions on anything that's coming up so far? Okay. All righty. So if not, we'll go ahead and finish up with chapter 9 here. So chapter 9.5 is the last part of chapter 9. And so this is going to cover how we are actually able to regulate genes. Okay. So in our last lecture, we looked at how we were able to produce various proteins. We were looking at how we were able to transcribe specific genes, okay, undergoing transcription, okay? And then how we're able to take the mRNA that we produce and then produce the corresponding protein through translation. Okay? So for example, if we need to produce the protein like hemoglobin, for example, right? There's going to be a specific gene in our DNA that we are going to activate. Okay? We're going to undergo transcription of that gene. Okay? Once we transcribe that gene into mRNA, we're then going to translate that gene to help us produce the hemoglobin protein. Okay? Same thing with any of the proteins involved or enzymes involved with cellular respiration. Okay? To help us go from having glucose to helping us produce ATP at the end of that process. Okay? Same thing if we want to produce melanin. So melanin is going to be another protein that we're going to want to produce to help us, get darker or help us produce dark skin. Okay? So those are all different genes that need to be activated. Okay? In order to, again, to activate those genes, you have to transcribe that corresponding gene in the DNA, okay? And then translate it into a protein. Okay? So we looked at how we were able to do that last week, but we haven't looked at how we're going to be able to actually regulate what genes are activated at any given point in time. If that's going to be what we're doing today. And we're going to be looking at how we're going to regulate gene expression. Okay? And so for any cell, okay, cells have to be able to make proteins. Okay? They have to be able to make the appropriate protein at the appropriate time. Okay? There's a time and place for each protein or any protein synthesis. And each cell needs a series of proteins to help it undergo its daily functions. Okay? However, you don't want to be just constantly producing the same protein over and over and over and over and over, every day, all day for the rest of your life. Okay? You want everything to be done in moderation. Okay? So we have to be able to regulate transcription and translation. Okay? So these are two processes that need to be regulated and need to be done in a controlled manner. Okay? Again, you don't want to be producing all these things just constantly without any regulation. Okay? So the process of either of turning on a gene or turning off a gene is what's called gene expression. Okay? So gene expression is how we're able to turn on a particular gene, okay, to help us produce some RNA, and then from there use the RNA to help us make some proteins. Okay? So what's going to be important when we're producing or when we're undergoing gene expression is we want to regulate when a gene is expressed, okay, when we actually activate it. Okay? We want to regulate how much of that protein we produce, okay? And we want to regulate when we're going to stop producing that protein. Okay? All three of those are going to be really important to do on a day-to-day basis, really on a minute-to-minute basis. Okay? We want to be able to regulate everything that's happening within that gene. It regulates its expression, regulate how much protein we're making, and then when to stop producing that protein. Okay? Again, we want to do everything in moderation. We don't want to have too much of a particular protein, and we don't want to have too little of a particular protein. Okay? We want to have the right amount to help us produce the functions that we need to be able to do, okay? Or that our cells need to be able to do. Okay. All right, cool. Alrighty, so when we look at the different types of cells that we're going to have, okay, each cell is going to have the same complement of DNA. Okay? So the DNA that's in your muscle cells, for example, is going to be the same DNA that's going to be in the skin cells, and the same DNA that's going to be inside your neurons, okay, or your brain cells. Okay. Now, if it all has the same DNA, how is it that all of these cells look differently and are able to have different functions? Okay. Well, the reason for that is because depending on the cell that we're looking at, there's going to be specific genes that are going to be activated to help that cell do what it's designed to do. Okay. So for example, the muscle cells are going to have specific genes that are going to be activated to help it be able to contract and help with movement. Okay. Skin cells are going to have a completely different set of DNA that gets activated, a completely set of genes that get activated to help you just produce your skin cells, to help produce like protection for the body. Okay. And the neurons, for example, your brain cells, they're going to have a completely different set of genes that are going to get activated, okay? And that's going to cause those cells to look a lot different and be able to produce electrical impulses throughout the brain to help produce communication between brain cells, okay? So even though they have the same overall DNA from one cell to the next, okay, each one has a specific set of genes that gets activated to make sure, or to kind of determine what that cell is going to be able to look like and what it's going to be able to do, okay? So it's going to make it specialized for each individual function. Okay? Does that make sense? Everyone kind of following me so far? Yeah, okay. All right, cool. Okay, so each cell has the ability to either activate or deactivate genes in response to the environment. And that's going to be really important because our cells, one of the important things about life or about anything that's living is it needs to be able to respond to its environment. Okay. Now, if a cell is not able to respond to its environment, that can possibly be pretty detrimental or lethal to the cell. Okay. So for example, if we're out in the sun for long periods of time, let's say we're out at the beach, we're just kind of chilling out in the sun, it's a very hot, sunny day, and the sun is just beating down on our skin, right? Well, our cells need to be able to respond to that ultraviolet radiation that the sun is releasing. Okay? And so what our skin cells are going to be able to do is it's going to start producing a protein called melanin. Okay? And so what we're going to do is we're going to activate the gene responsible for producing melanin. Okay? We're going to activate that gene. We're going to undergo translation of that mRNA to help us produce that protein melanin. Okay? And from there, that melanin that we produce will help protect our skin cells and then also create kind of a darker skin color. Okay, that's essentially how we tan, okay? Or how you guys tan, not so much me because I'm pretty dark. But that's pretty much how things work. Okay, if your skin cells were not able to produce melanin in order to protect yourself from that sun, then you'd end up being exposed to. You could potentially get skin cancer, or you can end up getting sunburns and things like that. And again, that's going to be due to either not being able to produce melanin fast enough or not being able to produce it at all in response to a change in your environment. Okay? So you have to be able to activate or deactivate different genes depending on the environment that you're put in. Kimberly says we go beet red first. Yeah, in some cases you do. Yeah. Okay. So that's how gene expression is going to be working. It's going to be a really important process for survival in certain instances. So gene expression is going to be happening a little bit differently in prokaryotes versus in eukaryotes. Okay? So in prokaryotes, transcription and translation are going to be happening pretty much at the same time. Okay? And the reason for that is because in prokaryotes, they're not going to have a nucleus. Okay? So if you remember in eukaryotes, transcription happens inside the nucleus. Because prokaryotes do not have a nucleus, okay, transcription and translation are both going to occur in the cytoplasm. And so they're pretty much going to occur simultaneously. Okay. So if prokaryotes need to regulate gene expression, okay, their gene expression is typically going to be only regulated during transcription. Okay. So if you can regulate what's happening during transcription, then you're by default going to be regulating translation because they're going to be happening at the same time. Okay. So if a prokaryote needs to increase the amount of protein that it needs inside of itself, it's going to create more transcription. Okay? If it needs less protein, it's going to start inhibiting transcription. Okay? So it's essentially going to be regulating gene expression solely through transcription. Okay, again, because they're going to be happening, transcription and translation are going to be happening at the same time. So here we have an example. So an example of gene expression that happens inside E. coli, okay, a bacteria called E. coli, is a region of the DNA called the lac operon. Okay. So the lac operon is a series of 3 genes kind of all adjacent to one another. Okay, we have the lac Z, lac Y, and the lac A genes. Okay. And so these are all genes that are going to be useful for helping E. coli be able to metabolize and absorb lactose. So in order for lactose to be used up by the E. coli, it needs these genes to become activated. Okay. So if lactose is not present in E. coli's environment, then the lac genes are not going to be transcribed, okay? Or they're going to be transcribed in very, very small amounts. But typically, they're not really going to be transcribed all that much. Okay. That's if lactose is not present in the environment. Okay. However, if we introduce lactose to E. coli's environment, okay, then these lac genes, lac Z, lac Y, and lac A, okay, those are all going to begin being transcribed. Okay. Once these genes are transcribed, okay, then the E. coli will have all the proteins necessary to help it break down lactose and use it as a food source. Okay. So here's how this whole operation works. Okay. So starting with when it's inactive. Okay, so when our lac operand is inactive, what's going to happen is we are going to have, or again, we're going to have a promoter region, and that's going to be where RNA polymerase is going to bind. We're going to have an operator region, and then we're going to have our lac genes. Okay. So if we do not have any lactose in the environment, and then the lac operand is going to be inactive. Okay. The reason it's going to become inactive is because we're going to have a protein called a repressor, and the repressor is going to bind onto a region on the DNA called the operator, okay, the operator region. And so what happens here is the repressor will physically prevent the RNA polymerase from binding and prevents it from moving forward. Okay. So it basically stands in the way of RNA polymerase. Okay. So if RNA polymerase cannot get past this repressor, it's not going to be able to transcribe these genes here. Okay. And if you cannot transcribe these genes, okay, then you're not going to be able to produce the proteins involved with breaking down lactose. So E. coli is not going to be able to use lactose in any way. Okay. So that's going to be what happens when lactose is not present, or when lactose production is or lactose metabolism is inhibited. Okay. The repressor binds to the operator region and then prevents RNA polymerase from transcribing these lac genes. Okay. So you block transcription. Okay. Now, when the lac operand is active, what we'll see here is that RNA polymerase will bind to the promoter region, and then it's going to begin to undergo transcription. Okay. So what's going to happen is RNA polymerase is going to undergo transcription here. We're going to start being able to break down lactose. Okay. E. coli is going to start being able to use lactose. And when it breaks down lactose, it's going to create a little product of the lactose breakdown called allolactose. Okay. This is a product of lactose breakdown. And what allolactose will do is it'll bind onto the repressor and prevent the repressor from binding onto the operator region. Okay. So because the allolactose prevents the repressor from binding on, a RNA polymerase is going to have a free rein to transcribe these genes. Okay. Okay. So if we're able to transcribe the LAC-Z, LAC-Y, and LAC-A genes, okay, you're going to be able to produce the protein that helps you helps E. coli break down lactose. And then lactose metabolism is going to be allowed. Okay. So that's how prokaryotes are typically going to have their gene expression be regulated. Okay, there's going to be a repressor region, or a repressor protein that will bind onto the operator region in order to prevent that gene from being activated. Okay. If we do want that gene to become activated, the repressor is going to be moved away, and the RNA polymerase will be able to transcribe all of these genes. Okay. Any questions on what's happening with prokaryotic gene expression? Okay. Does this make sense? If it makes sense, go ahead and throw a check mark or a star or say something so I have an idea of where we're at with this. Any questions? All right, looks like we're okay. All right, cool. So question here. So where do transcription and translation take place in eukaryotes? Okay. So we have A, transcription occurs in the rough endoplasmic reticulum. Translation happens in the Golgi apparatus. B, transcription happens in the mitochondria and translation happens in the rough ER. C, transcription happens in the nucleus and then translation happens in the cytoplasm. Or D, transcription occurs in the smooth ER and translation occurs in the nucleus. Which one is correct? Yes, so I'm seeing C, and C is correct. Perfect. All right, so in eukaryotes, transcription is going to happen inside the nucleus, and then translation will take place inside the cytoplasm. Good. Okay, so this is our last slide, and this is going to be basically how eukaryotes are going to regulate gene expression. Okay. So in eukaryotes, transcription and translation are going to be happening in different locations. Okay? So in transcription is going to be happening inside the nucleus. Translation is going to be happening inside the cytoplasm. Okay? And so that's going to be different from what we see in prokaryotes, where everything is going to be happening in the cytoplasm. Okay? Here we have a separation of these two processes. Okay? And so they're going to be separated by the nuclear envelope. Okay? Now, because we have this separation here, we're going to be able to regulate gene expression at multiple different points, okay, of our DNA here. Okay? The first place where we can regulate gene expression is going to be within the DNA itself. Okay? So this is called the epigenetic level. Okay? So one way that we can regulate gene expression is when our DNA is condensed or whether it's less condensed. Okay? So remember when we were looking at last week, we were looking at heterochromatin, where you have the DNA being very, very condensed around the nucleosomes, and then euchromatin, where the DNA gets kind of loosened up around the nucleosomes. Okay. So in heterochromatin, because everything is so condensed, there's not very much room for any of the enzymes to come in and begin transcription. Okay. And so in heterochromatin, because there's so much condensing, okay, there's going to be very little gene expression happening in heterochromatin. Okay. Now, when it becomes loosened up and as euchromatin, and that's going to allow more room for those enzymes to come in. And so we're going to have more, or a higher level of gene expression happening in euchromatin. Okay. So that's what we call the epigenetic level, okay, whether it's condensed, where the DNA is condensed, or less condensed. Okay. That's going to be the first level where we're going to be able to regulate gene expression. Okay. The second place where we can regulate gene expression is going to be at the transcription level, okay, when you're actually transcribing the DNA into mRNA. Okay. So as we add those particular enzymes to help us begin transcription, that is going to be another place where we're going to be able to regulate gene expression, okay, specifically during transcription. Okay. The third place is going to be at the post-transcriptional level, and so that is going to be where the the RNA that we produced gets processed. Okay. So when we're undergoing splicing of our mRNA, when we're undergoing the addition of the 5' cap and the poly A tail, okay, those are all going to be places where we're going to be able to regulate our gene expression as well. Okay. The fourth place is going to be at translation. Okay, so when we're actually producing the protein, and that is going to be the fourth place where we're going to be able to see gene expression. And the last place where we're going to see gene expression is going to be at the post-translational level, okay, after you produce your protein. Okay. So after we produce the protein, that protein can be modified. And so it can be modified in such a way that makes it more active. It can be modified in such a way that it becomes less active or inactive altogether. Or in some cases, it can be modified in such a way that it can either turn off or turn on, depending on different things in its environment. Okay. So gene expression in eukaryotes can happen at multiple different levels of this entire process. Okay. At the epigenetic level, okay, where your DNA is either going to be coiled very tightly or less tightly coiled, okay, around the nucleosomes. Okay. It can happen at transcription. It can happen during the post-transcription level where we're processing our RNA. It can happen during translation, and then it can happen at the post-translation level where we modify the protein that we produced after translation. Okay. All right, and that is everything with this chapter. Any questions on chapter 9.5? Okay. All right, so if there are no questions, there is some classwork that's been posted, so the classwork is relatively short. There is also a very short homework assignment, so it's only about 4 questions. That will be due on Thursday, this upcoming Thursday, so 2 days from now it'll be due. And so that'll give you kind of an indication of what kind of questions to expect from chapter 9.5 on the exam. Okay. So on Thursday, what we'll do is we will take some time to go over the homework on Thursday. That should take, maybe 10, 15 minutes max. And then if there are any questions before you take the exam, I'll be available to answer any questions for you. And then, yeah, that'll be it. And then you go ahead and take the exam. Okay. So remember, exam 3 will be this Thursday as well. Okay, so we'll have a short little lecture just to kind of go over the homework, and then we'll have the exam. And again, you can take that exam anytime between 8 a.m. and 11:59 p.m. Okay. We get to these questions. So there is lab today. So if you're in the Tuesday lab or the Thursday lab, we do have lab this week, and we'll have lab for the rest of the semester, okay, up until finals week. And then again, if you haven't already done the new classwork from last week, new classwork for 421 and new classwork for 423, make sure you get that done as soon as possible. And then, yeah, that's pretty much everything. So yes, Paul, this is the last chapter before the exam. Yes. Any other questions that I can answer? Oh, so for the final, there will be 2 more chapters. We'll be going over chapter 11 next week, and then chapter 19 the week after, and then we'll have finals week after that. Okay. But again, we'll get there next week. Cool. Any other questions? Okay, so if there's no questions, you guys are free to go. Make sure you take care of that classwork, and then make sure you knock out the homework before Thursday. So Jared's question, will the final cover all the topics from the entire semester? Yes, it will cover all the topics from this entire semester. So it will be cumulative. Cool. Okay, and I'll talk more about that